the polysaccharides that we're going to talk about. So when we are linking many monosaccharides together, cellulose is going to be a long chain of polymers, 2,800 D-glucoses. And it's the beta-1,4 glycosidic linkages, just like in cellulose. And again, because we don't have the enzyme that can break apart this beta-1,4, cows have the bacteria that can do that. Why is cellulose good for structure? Because we have a lot of interchain and intra-chain hydrogen bonding. So when you are chewing bark, like my son when he was 4 years old, would go up to trees and take bark off and chew it. He's 9 now, so he doesn't do that anymore. But when he was 4, that was his favorite hobby. And he would go to trees and chew the bark. And so what he was doing with his teeth is breaking apart these hydrogen bonds. I'm glad he's over that. Right? But again, then we can't break the bonds that will hydrolyze it down to glucose. Starch is, again, this is supposed to be alpha right here. There's a few different forms of, different forms of starch. A straight chain is just unbranched of the alpha D-glucose, joined only by the alpha-1,4 glycosidic bonds. This is supposed to be alpha. And so we can see this in the next slide. Amylopectin, though, is actually the same thing. It's only glucose. It is, again, the alpha-1,4 glycosidic bonds, but we have branches that are alpha-1,6. And we have different enzymes that will break up the different kinds of bonds. Amylases, ASE is for the enzymes, OSE is for the sugars, for the endings. So this amylase will hydrolyze the alpha-1,4, and debranching enzymes hydrolyze the alpha-1,6. So all these question marks on this one are alpha. And I did fix it this morning, but it didn't stick. Anyway, I'm sorry. Let's take a look at this. So this is the difference. The amylose is alpha-1,4 glucoses, straight chain. Amylopectin has the alpha-1,4 I'm showing down here and up here, although occasionally we have an alpha-1,6. These are branches. Now I say that this is a straight chain. On this straight chain, how many reducing ends do we have? One, right? So one at the end, wherever it stops. How many non-reducing ends do we have on this amylose? One, right? We have one non-reducing end and one reducing end. On amylopectin, how many reducing ends do we have? One. One. How many, in this one, how many non-reducing ends do we have? Two. Okay. And there's more branches. Okay. So if you look back here, there's a lot of, it's branched a lot. So we have a lot of non-reducing ends. So we said that amylose was a straight chain. Really, it's not straight. It forms a coil. And at your next party, you should go to a CVS and get a tincture. It's a bottle of tincture of iodine. And then you should drop the iodine on all the food. And then you can point out to your friends which foods contain starch and which ones don't. Because the iodine molecules will get trapped within the amylose starch and turn from purple to black, right? So they might kick you out of the party. But this is something I might do at my next party. So isn't that cool? Look how big iodine is to carbon. Isn't that weird? We use this in analytical chemistry as an indicator. The starch will be an indicator for free iodine. So it goes from, if you have free iodine, it's like totally black. And the second that the free iodine goes away, it goes clear. Isn't that fun, guys? It's like the best one. So cool. Anyway, glycogen is also a starch. However, and it's the same thing as our amylopectin, except that it's more frequent branching. every fourth or every eighth residue. So it is more highly branched. So amylose is just the 1,4, alpha 1,4 linkage. Amylopectin is alpha 1,4 and alpha 1,6 to give more non-reducing ends. Glycogen is the same as amylopectin, except it has more frequent branching. It's stored in the liver and muscle cells. And the way we have our sugars is the reason why we have the more non-reducing ends is because the enzymes to free the sugar to go through metabolism take from the non-reducing ends. So the more non-reducing ends we have, the faster mobilization we can get. So if we just have amylose, we have to chomp. We only have one place to chomp, okay, to get the glucoses off to get it through glycolysis. However, if we are storing glycogen, we have lots and lots of non-reducing ends. So there's lots of places for the enzyme to chomp the glucose off. Glycogen, is anybody a long-distance runner? Yeah. How long does the glycogen last when you run? It lasts, for me, about an hour 15 minutes. When you run, you usually have about an hour and 15 minutes of glycogen in your muscles. And that's why, do you ever see the goo stuff? That's what people are, when they eat the goo. I do gummy bears, and I just hold them in my gums to just give a little bit of sugar so that I can get, so that means you've used up all the glycogen in your muscles. That's a good thing. But that's
for an hour and a half.
Yeah.
So
Yep.
it'd be different for like other disciplines.
If you're
Faster.
if you're running faster, but at a normal pace, it's, I think, yeah. For me, it's an hour and 15 minutes I hit the wall. I don't know. Maybe better people can store more glycogen. I have no idea. But for me, it's an hour and 15 minutes. I'm like, I'm done. So, but then you get over it when it starts to get it starts to go for fat. So that's what you're doing. When you only run for a half hour, you're just using up a little bit of your glycogen. But it's good to, get some of it. But why do we store glucose as glycogen? Why are we doing it? Not only just to get more non-reducing ends, we have to actually expend energy to get glucose molecules in there. And so don't look at the answer, but why do you think? The answer's in your notes, but why would you think? Well, one thing about the glycogen molecule is that it's so compact that it's not soluble. So it's insoluble, and it takes it out of creating osmotic pressure inside the cell. If you had all those sucrose or glucose molecules dissolved in your cell, it would burst the cell because of osmotic pressure. So it keeps it insoluble, so it doesn't add to that effect. All right, I'm just going to go quickly through other polysaccharides that I don't really care about. So here, chitin is some hard stuff that when you step on a bug, it goes crack and crunch, right? So, like does anybody like to eat lobster? No? Come on, lobster's great. Anyway, chitin is a repeating disaccharide of, N-acetylglucosamine, and it really produces like an exoskeleton because of some hydrogen bonding. And this is supposed to be beta and beta. I apologize. Beta and beta here. So it's just like cellulose in plants. The chitin is just like our cellulose in our plants. In bacterial cell walls, they have heteropolysaccharides. What we mean is that you have repeating units of what we call NAM and NAG. So these guys are crazy. They have the nitrogen on there, and these crazy groups here. And then look how crazy that gets. That's unbelievable. But what happens is, and the reason why bacterial cell walls are so strong is because we are cross-linking So here is our sugar chains, and they are cross-linked with amino acids. That is super, super strong. It's very, very hard to open up a bacterial cell wall because of this. So this is a good question that I might ask at the end of the, I don't know, why are bacterial cell walls so strong? You would answer, you not only have the polysaccharide chain, but it's cross-linked with amino acids. Plant cell walls is mostly cellulose, right? Like that, but they also contain pectin, and that's alpha right here. This is alpha. So pectin draws water in. It has an acid here. This is the bottom. This is carbon number 6 that has turned into an acid, so it's a uronic acid. This is a gelling agent in your yogurt and jams, okay? So this is, and it contains the goo in between the plant cells. The plant cell walls also have lignin that is a polymer of an alcohol. All right, so I just quickly went over theirs. I told you the one thing that I really wanted you to know. Glycosaminoglycans are a group of polysaccharides that play a role in cell functions. You have disaccharides repeating where there's an amino group and the other has a negative charge. A few examples are heparin, hyaluronic acid, chondroitin sulfate, right? Connective tissue. That's why people might take supplements of that. And so they're relatively inflexible, these glycosaminoglycans, and very charged. So look at the name. So glycan, it means polysaccharide, right? Amino means amino acid. So you know that there's some sort of combination. So the amino sugar, that has the negative charge and it has a sulfate or a carboxylate group, and that will draw water towards it. So your connective tissue needs that water. And of course the hyaluronic acid, that's in the goo in your eye, lubricating our fluids of the joints. Glycoproteins, I already talked about that. Just remember, okay, so we take a look at this. Glycans is the last word, so that means there's more sugar than amino, right? Here, glycoproteins, there's more protein than sugar. So the glyco, that means sugar, and the protein, obviously we know is amino acids. So here we have more protein. So whatever's at the end, we have more of. So here, antibodies are glycoproteins. I already talked about how I would characterize the short chains. And they're the antigenic determinants. And blood groups were the first ones to look at it, to see that we saw were the sugars identified were part of the recognition. And so we have AB, or O, depending on the sugar on the surface of our red blood cells. And so we look at the terminal monosaccharide, and that determines on the non-reducing end, and that determines your blood group. So we attach it at the end. So here, these are, don't worry about this part. I just wanted you to see if you are type A, then at the end you have a beta N acetylglycosamine. And then if you are a B, you have a galactose. So that's recognition. So the take-home part of this, we make our polysaccharides, our disaccharides, but we also use them for recognition.